All right, so we're going to open up chapter 3, which is going to be about the biomolecules unit, or the biomolecules of life. And if you were in regular bio or PAP bio recently, you'll probably remember the 4 categories of biomolecules. So for the do now or entrance, I kind of just want you guys to think about this. I want you to try to write down, list 3 biomolecules found in your food that provide energy. There's 2 that provide food more often, and I'll give you a hint. 1 provides short-term energy, 1 provides long-term stored energy, and then 1 is used as a last-ditch resource for energy, but it's usually used for building structures in your body. And then identify the last one that does not break down to produce energy, and this one usually stores important information in your body. Right? So our learning objective is that learners will be able to justify the selection of data regarding the types of molecules an animal, plant, or bacterium taken as building blocks or excrete as waste. The agenda is biomolecules introductions review notes. So we're going to go over the introduction and vocabulary that you need to talk about all of biomolecules. And then for the rest of this week, we'll actually do the notes on the 4 types of biomolecules. And then your homework, again, remember, this one's going to be a little bit of a short recording. So once you have extra time, make sure that you finish up your chapter 3 summary and learn smart, because that's due on Wednesday before class, which means technically you should have it done by Tuesday night. So now back to the do now. So the 3 biomolecules used for energy are, first of all, carbohydrates, right, short-term energy. Fats and lipids, same thing. They are the long-term storage energy. And then proteins are the ones that are not preferred for energy, but can be used as a last-ditch effort. So if you ran out of carbs and you ran out of fats, and your body will actually start breaking down your muscles and some of your bones to, they'll slowly pull it out of your muscles and bones to use for energy if you don't have anything else, right? And you need to continue surviving. Biomolecule, the single biomolecule that does not provide energy, no matter how much of it you have, is nucleic acids. And that's usually your DNA and your RNA. So like, you shouldn't be breaking that down regardless, and your body makes sure that it doesn't. So let's talk about chapter 3. So we'll talk about organic versus inorganic molecules first, right? So this vocab is kind of important because organic molecules are ones that have carbon and hydrogen in them. And most of the things that are building blocks of life are organic molecules because they contain carbon and hydrogen, right? They're the structural molecules that all life is made out of. Now there's also inorganic molecules. So they either have carbon or they have hydrogen, but they contain other elements, like they'll contain iron, right? Hemoglobin in your blood contains iron because it helps to hold on to oxygen. They'll contain there are parts of your body that contain sulfur, right? In your hair, it actually determines the texture of your hair, right? I have very straight Asian hair, so my sulfur bonds in my hair are different from somebody who has curly hair, right? And then there's like phosphorus, there's gold, there's not that much silver. There's a lot of other things in your body that contain like other elements. So those are inorganic molecules. So the 4 categories of biomolecules that we mentioned during the do now are carbohydrates, lipids, proteins, and nucleic acids. And we'll talk about those in more detail later this week. Functional groups are basically chemical groups that are attached to any of the above 4 molecules, right? They're a specific combination of bonded atoms, and we call them functional groups because they all have specific functions. So they always react in the same way, so we can categorize them and make it super easy. And when a molecule has a functional group, then we know the job of the molecule because the functional group, again, always has the same job. It always reacts in the same way, regardless of the particular carbon skeleton, right? The carbon skeleton that's on the main molecule actually isn't going to affect its job that much. Like, we already know the general job from it being in whatever of these 4 categories, but the functional group tells us how it interacts with other chemicals or molecules, what it's going to do in your body when it reacts with them. So it means that a biomolecule's chemical behavior is due to the functional groups attached to it rather than the carbon skeleton that they are attached to. So we usually, this is in red because we usually refer to functional groups as R groups in general. And so whatever R group is attached to your carbohydrate or to your lipid or to your protein or to your nucleic acid determines its specific job. So there are a couple of groups over here, like hydroxyl groups, right? We talked about that. You should recognize it from when we're talking about pH, right? Because OH is a hydroxide and it's a base, right? So when you have a hydroxyl added on, the compound attached, it actually turns the entire compound into an alcohol. Okay, so ethanol, right, is a common alcohol, isopropyl alcohol. It all has these OHs attached to their chemical groups. And it's important because it's polar and it forms hydrogen bonds, because the difference between the O and the H, right, the O is a little bit more negative and the H is a little bit more positive. And you will find them a lot in sugars and some amino acids. So you'll find them mainly in the carbohydrates and the proteins groups. Another one is a carbonyl, right? So it has the O, it looks like it has an OH group, but the O and the H actually aren't connected, right? They're both connected to the same carbon, but they're not connected to each other. And this changes its job, right? So it becomes aldehydes if it has a carbonyl. And an example is formaldehyde, like when they store dead bodies and they want to preserve them, like in the jars, like you see in movies, or even just like the dissections that you do in school, those are preserved in formaldehyde and makes things stop rotting. And again, they're special because they are polar and they're present in sugars, right? So you'll find them in this carbohydrates group. You can also have an O attached to the carbon, but then it's attached to a different group. And that makes it a ketone, like acetone. So if you ever like wore nail polish before and you need to get it off, acetone is nail polish remover. And acetone is special because it's polar and it's usually present in sugar. So it's usually things that are attached to carbohydrates, right? There's a bunch of other groups too, and they're all really interesting because they all have their own special jobs. So you can pause and read over these. I don't need you to write them all down, but it's good that you kind of just get to read over them, because if I ever mention them, then you kind of know or have an idea of like, what is its job or what category it's in then. And some of them are kind of obvious, right? Amino is going to be an amino acid, right? So it's going to be protein related. Sulfhydryl has sulfur in it. So if you see an S in it, that's a sulfur, right? Phosphate groups have a P in it because it's a phosphate or a phosphorus, okay? Some of them are pretty obvious, but some of them are not as obvious, like the ones up top, but they're all really, really common because these are the most common ones. Okay, so let's go into more vocab now. So monomer, you should have learned this before. Monomer is the smallest subunit of biomolecules. And a polymer is the largest of biomolecules. So when you take a bunch of monomers and link them together, then you get a polymer. So polymer is just a chain. You take the same thing and you link it over and over and over and over and over and over again, and then you get a polymer, right? Poly means many, mono means one. So examples are, if you have carbohydrates, the subunit or monomer name for that is a monosaccharide. Mono, one, saccharide, sugar, one sugar, monosaccharide. Then your polymer is going to be a polysaccharide or many sugars, polysaccharide. For lipids, you don't actually have a true polymer. They don't really link, they actually stack. So that's why this is just very general over here. So you have one glycerol attached to 3 fatty acids, and that's your subunit, that's your monomer of lipids. And since we don't really have a true polymer because they never really link, it's called fats here, but they just stack on top of each other, right? In oils, they don't really stack because they haven't like solidified, but if you cool it down enough, the oils will also stack and become solid. Proteins, we have amino acids. And when you have a lot of them together, you have polypeptides, right? Poly is many, and then peptide is the word for a protein chain, right? Because you can get amino acids that form peptides when they're like 10, around 10 combined together in a chain. And you get polypeptides, which are a really, really long chain, up to like hundreds of peptides together, which are polypeptides. And then you have nucleic acids, which are nucleotides, right? Each individual nucleotide has a phosphate group, it has the carbon sugar, and then it has an N group, well, a nitrogen group. And so all of those 3 make up a nucleotide. And then when you get them in chains, you get your DNA or you get the RNA that you have in your body. Alrighty, so the first thing that we're going to talk about of how you even get from a polymer to a monomer or a monomer to a polymer. So the first process that we'll talk about is called dehydration synthesis. And let me move myself out of the way so that you can see. The process of removing an OH or hydroxyl and an H or hydrogen atom, which is the equivalent of a water, and you use that to join 2 subunits together. So joining 2 subunits means that you're actually combining them. So that means you're starting with 2 separate pieces, 2 monomers. And when you do dehydration synthesis, then they become a chain, right? You see the 2 monomers still, and they're connected by a chain. And when you're doing the connection, a water molecule pops out and leaves. So just to explain the name, the name actually explains the process. It's kind of cool. So dehydration, right? If you're dehydrated, you're lacking water, right? Water got taken away. So when you dehydrate, you're going to take the water away in this process, right? Because you took an OH and you took an H. And when you combine that together, that becomes H2O, because there's two H's and there's one O. So you dehydrate by taking the water away. And synthesis is a word where, for example, if you are synthesizing drugs, you're making drugs, right? So you're taking away water, dehydration, synthesis to make a polymer. Dehydration synthesis, taking away water to make a polymer. And so you see that written over here, kind of in bad handwriting, my bad. And then you see this in this picture. So you have a monomer, and one of them is exposing the OH, the hydroxyl group, and one of them is exposing their hydrogen. For example, this monomer would also have a hydrogen attached somewhere else, and this one would have a hydroxyl group attached somewhere else. It's just these are going to directly interact. And when you pull them both off, they'll combine into a water molecule, and that'll leave. And then that creates a bond that links the two of them, because now they have this empty space where their OH used to be, and then they have an empty space where the H used to be. And they'll actually make that linkage right here, and now it becomes a chain. This is now going on its way to become a polymer. It's only a dimer right now. Remember, di means two. It's a dimer right now because you combined one monomer with one monomer. But if you combine more and more and more and more and more through this process of dehydration synthesis, then you can get a polymer. Now, we also have this process called hydrolysis degradation. We usually just call it hydrolysis. And so if you break it into its words, hydro and lysis. Hydro means water, right? Hydro, hydration, getting water. Lysis, if you think all the way back to hypertonic and hypotonic and learning about tonicity with like solutions, then you'll think of lysis is when a cell bursts or breaks. So basically the name just means we're gonna use water to break. Hydrolysis, water break, okay? And the term degradation just means it's breaking down, but we usually don't add this term in. It's usually just called hydrolysis. So the process of adding an OH from water to one subunit and an H to the other subunit to break them apart. And these are opposite processes, right? So if you look at this diagram, it's literally the exact opposite. So if you start off with your monomers attached together in a polymer, and you add the OH and the H from an H2O by breaking the water apart, then it actually separates this bond that used to be between them. And now they break apart because there's no bond connecting them anymore. So you've actually broken the chain by doing this. So again, these are opposite processes, but they're important because in metabolism, we spend a lot of time making and breaking these polymers because either we need to use the energy, so we break them, or we're building them because we're storing energy, so we make the bonds. Okay, so what's important is that enzymes are a molecule that speeds up a reaction by bringing reactants together. So for example, if you're trying to, get in a relationship with somebody and you have your friend wingman you, or if you have your friend introduce you, that's essentially what the enzymes are doing, right? If you wouldn't have met them easily just by like random chance, but if you have somebody introduce you to each other, then it's more likely and more quickly that you'll start a relationship if you want to. So an enzyme is a molecule that speeds up a reaction by bringing reactants together and helps them form new molecules. The enzyme remains unchanged during this process. So like your friend's not really affected by your relationship because it's not like they're entering one, right? So here, an enzyme would be speeding up the hydrolysis by bringing the water and breaking the water apart to help add them on. Or up here where you have the enzyme helping hold these in place so that the water parts, the parts that are going to become water, easily touch and break apart. And then the enzyme is also holding the monomers in place while the bond between them forms to make into a polymer. So enzymes are important because a lot of the times when you have processes, you can't actually do them at a speed that is necessary for survival without enzymes. There are so many enzymes in your body, and these enzymes are actually protein molecules, and they remain unchanged. So once they're made, they're there, they're good to go, they're usable, reusable, and that's really important. Alrighty, so this is the end of notes for today. I told you it wasn't gonna be too long. I know I talked a lot, but the actual content's not that long. You're going to move on now to, if you have extra time during this class period, please finish up your chapter 3 summary and your Learn Smart on time, which means before Wednesday's class, because it's hard for me to grade them on McGraw-Hill if it's late. since it doesn't show it up, and you'd have to submit it for me then to see, which is another step for you to do. So get your work done and make sure that you turn it in on time. You can turn in these notes too for extra credit underneath here.